Hello, ladies and gentlemen. Thanks for tuning in to our webcast today. I hope your week is great so far. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix. And today, Nathan Fortier is a Senior Product Engineer and Field Application Scientist with us at Golden Helix. And he's going to give us an in-depth look at the new CNV analysis capabilities in VARSeq. Nathan, take her away.
So today, what I'm going to be talking about, as Cheryl mentioned, is CNV analysis in VARSeq. So we're going to start out and just go over a quick overview of Golden Helix and who we are as a company. Next, we're going to talk about the motivation for calling CNVs on NGS data as opposed to other methods that are currently out there. After that, we're going to describe our method and demonstrate the way the method works within the software. Then finally, we'll answer any questions and discuss availability and our roadmap. If you have any questions during the presentation, please feel free to use the questions pane in your GoToMeeting window and enter any questions that you have there. So for those of you that don't know, Golden Helix is a global bioinformatics company. We were founded in 1998. We spun off from early work with GlaxoSmithKline, who was a key investor in the company. Our first initial product was a product called Helix Tree, which has now evolved into the SNP and Variation Suite, or more commonly referred to as SVS. We've since added two new products, Genome Browse and VARSeq. The company has 17 years of expertise in the field of bioinformatics. We've been in this market since before GWAS was even born. We have a large number of customers all over the world. We serve over 300 organizations, including top-tier research universities around the world, major government institutions, and pharmaceutical companies. In fact, we have customers on every continent except Antarctica. And we're proud to say we have more than 7,000 installs of our product. We've also been cited in a large number of peer-reviewed publications. We've been cited in over 1,000 publications in prestigious journals such as Science, Nature, and Nature Genetics. Here at Golden Helix, we are known for our quality products, excellent customer service, and domain experience. We've been developing software for over 17 years, and we have a wide range of talented experts on staff, including biostatisticians, mathematicians, and software engineers. Here at Golden Helix, we don't just sell products. We seek to understand our customers and their needs. And ultimately, for many of our customers, we become trusted advisors. We have a breadth of industry experience. Our service department has completed dozens of research projects for customers, and so we understand real-world research obstacles and how they can be overcome. We also attend top conferences in the industry, and we speak at leading roundtables at these conferences. And in this way, we really track the pulse of the industry. We provide a community of support with the ability to learn best practices from other researchers and share knowledge with peers. And we also sponsor events that help further genomic research. So when you buy a product from Golden Helix, you're not just getting a piece of software. Ultimately, we provide you with all the training and support that you will need. We have a team of experts that's available to provide the resources needed to help complete your analysis, from training to technical support. We support our customers at every step of the way. And we understand the ecosystem of our clients. And ultimately, we know that time is of the essence, especially in this industry. And so we strive for same-day business turnaround when contacting our support team. We're also proud to say that we are one of the most transparent companies out there. All of our major methods and algorithms are thoroughly documented. In many cases, this even includes the math and complete article citations. We completely shun the black box approach. And ultimately, I'm not aware of another competitor whose methods are as transparent as ours. All of our software is designed for your standard desktop or laptop. We really pride ourselves on the speed of our applications. And so ultimately, you can run this software without ever needing to access the cloud. And we eliminate the need to work with the biostatics core. And ultimately, our clients can usually get results within minutes. We also provide the ability to leverage insights quicker. We have a decisive edge in today's competitive market. We also are proud of the customizability of our software. We offer expandable and customizable platform via our scripting. In fact, all of our reports can be customized. Custom scripts can be used with our software. And we create many custom scripts and reports ourselves that can be downloaded from our website at no additional cost. Additionally, we're species agnostic, and so we work with data from all diploid organisms. This includes humans, plants, animals, all of it. So now that I've given a brief overview of our company and who we are, let's jump right into CNV detection and talk about current best practices and why calling CNVs from NGS data is advantageous. So some of you may know the current best practice for CNV detection is chromosomal microarrays. But detecting CNVs using this approach has a few setbacks. The most obvious setback is that if you use this approach, you're going to have to perform an additional assay. This will cause your analysis to take additional time, as performing a chromosomal microarray is fairly slow, but it's also an additional expense. And beyond that, chromosomal microarrays ultimately are only capable of detecting very large events. We're talking on the order of thousands of base pairs. And so this brings us to calling CNVs from NGS data. If we call CNVs from NGS data, ultimately we can take the coverage data that we pull from your BAM files, and then we can call CNVs from this coverage data. This allows us to get rid of the need to perform additional assays because you can run it on your already existing data. This saves time and this saves you money. But beyond that, performing CNV analysis on NGS data allows you to detect very small CNV events. We're talking on the order of 100 base pairs, single exon events. And events of this scale just simply cannot be detected using traditional chromosomal microarrays. So as I mentioned, in our approach, we're calling CNVs from your coverage data that's stored in your BAM files. And unfortunately, this data can be pretty noisy, and this presents some challenges when trying to call CNVs from this data. And this can really be exemplified by the figure on the right. So in this figure, what we're showing is the pile-ups. That is the actual read depth at each of these exons for 3 different samples: sample 11, sample 12, and sample 13. Specifically, we're looking at a number of exons in BRCA2. And there's a few things to notice here. First of all, the coverage varies pretty wildly between samples. You can see that sample 11, for example, has much lower coverage than sample 12 and 13. But beyond that, the coverage actually fluctuates pretty wildly between targets as well. Looking at any of these samples, we'll see that the coverage at one target is very different from the coverage at another target. And ultimately, the vast majority of these differences that we see here have nothing to do with CNVs whatsoever. Most of this fluctuation comes from either systematic biases in the data that are just artifacts of the approach that's used, and sometimes they are just there, just random artifacts that are just there by random chance. And so ultimately we need to find a way to deal with this noise. And it's worth pointing out, in this image, there's actually a CNV present in one of these samples. And I have yet to encounter anyone who, looking at this image, just looking at the raw coverage data, can actually pick out the presence of this event. And we'll get into actually which sample has this event later on once we look at the metrics that we use to detect these CNVs. But ultimately, our solution to addressing this noise is twofold. First, we perform data normalization so that the coverage profiles between the samples are actually comparable. Because as you can see, for example, sample 11 here has much lower coverage. And so by doing this normalization, we basically normalize out these differences between the samples over the entire coverage profile. The second thing we do is we compare each sample of interest to a collection of reference samples. And what this allows us to do is establish what a normal coverage profile looks like for each individual target. And once we have that baseline established by our reference samples, we can get a picture of what our sample of interest looks like in comparison, and from there we can see evidence of CNV events. So as I mentioned, we're ultimately calling CNVs using information derived from a collection of reference samples. Specifically, we use 3 metrics as evidence of a CNV event. The first 2 shown here are the most important metrics. That is the z-score and the ratio. These are the primary metrics that are used to detect CNV events. And these are the metrics that are derived from our collection of reference samples. The z-score just gives you information about how many standard deviations away your sample of interest is from the reference sample mean for a given target. So for example, if we had a z-score of 7, or say -7, that would indicate that we're 7 standard deviations away from our reference samples. The ratio provides us with a different set of information. The ratio is just the normalized coverage at a given target divided by the reference sample mean. And these ratios can give us information about specific kinds of events because there's specific values that we would expect this ratio to take on for certain specific kinds of events. For example, heterozygous deletion, you would expect about half the coverage at a given target if that target is in a heterozygous deletion. And so we would expect this ratio to be around 0.5 for this kind of event. Likewise, for a homozygous deletion, we would expect the ratio to be around 0. The last metric that we use is ultimately a supporting metric. It's not the primary metric used for calling CNVs, but it gives us some additional information, and that's variant allele frequency. And we'll get into the details of how that works a little bit later on. But ultimately, all these metrics are input into a probabilistic model. And this probabilistic model is what we use to call our CNV events. And you can see the benefit immediately of using these metrics, especially the ratio and z-score, if you look here on the right. Because looking at the raw coverage data, we cannot actually see the presence of this duplication that is shown here at the bottom. It's only when we introduce these metrics that we've computed relative to our reference samples that we can actually see the presence of this event, because we can see the spike here in the ratio and we can see the spike here in the z-score, indicating the presence of this duplication. So let's go ahead and look at the section of BRCA2 that we saw in the previous slide. So this is the same region of BRCA2. And again, we have the coverage for sample 13 shown here. But now we've added some additional data. Here at the bottom, we actually have our CNV state. So we can see that these 4 exons are a duplication. And here at the top, we have plotted the ratio and z-score, which is used as evidence to determine the presence of this duplication. And you can see that while before we had no way of seeing through the raw coverage data that there was an event here, through these metrics we now get clear evidence. You can see, the normal region here on the left, and then you can see this large spike in terms of ratio and z-score, indicating the presence of this duplication. And if you look at the specific values, you can see that we're anywhere between 3 and 9 standard deviations away from the normal. So it's really quite a large deviation from your reference samples. And your ratio here looks like exactly what we would expect to see for a copy number 3 duplication. We have a value of around 1.4, and ultimately the value we'd expect for a copy number 3 duplication would be 1.5. So we've got very clear evidence of this duplication thanks to our comparison against these reference samples. So if you recall, I mentioned that there's a third piece of evidence that we incorporate, and that's variant allele frequency. Again, this isn't a primary piece of evidence. You could run the CMV caller without any variance in your project, and chances are it would still perform pretty well. But variant allele frequency allows us to eliminate certain kinds of false positives, and it allows us to strengthen evidence for certain kinds of true positives. And this is because there are certain variant allele frequencies that we expect to see for certain kinds of events. For example, for heterozygous deletions, we expect we would only see variant allele frequencies of 0 or 1. And so if we see variant allele frequencies that deviate from this, this is evidence that there isn't a heterozygous deletion at that location. Likewise, for duplications, we expect values of 2/3 and 1/3. So that can, if we see values that are like that, that can provide us evidence that there is a duplication at a region. And you can see the usefulness of this metric in this figure. So the first plot we have here is just a plot of the variance overlapping a given region. Next, we have the actual states that were called for a number of different targets without using variant allele frequency. And so you'll notice that we've called these 2 targets as a deletion. Specifically, they were called as a heterozygous deletion. Now, once the allele frequency is incorporated, we see that there are variants here that have a value of 0.5, which should not be possible for a deletion event. And so these variants give us evidence that this is not a real deletion and allows us to correctly call this region as diploid. And so that's what this variant allele frequency gives us. Our algorithm also provides a number of quality control flags that allows you to identify either events that might be questionable or identify samples that might be problematic for CNV calling. We will flag an event if the event targets seem like they have fairly low coverage. If the coverage drops below a certain threshold, it becomes difficult to call CNVs reliably, and so we will warn you if a target has coverage that falls below that threshold. If there's high variation between samples, we will provide a warning. That is, if the coverage fluctuates really wildly from sample to sample in your set of controls. We'll also flag an event if it can't be differentiated from noise at a region. And this means that you see there are certain regions where you will see very large variation in coverage, much larger than you see on average in the rest of the genome or exome. And we could have a case where an event is called, but looking at the region, it really doesn't rise above this noise. And if that's the case, we'll flag the event and let you know. We also provide a number of sample level flags. We will, for example, flag a sample if it doesn't match the references. That is, if there's a very large % difference between your sample of interest and the reference samples, we'll flag that sample. We'll flag a sample if it has extremely low coverage, and we'll also flag samples if there's high variance across the target region. That is, if the coverage fluctuates too wildly, we'll flag the sample. So I'd like to take a moment to talk about reference samples, how reference samples are chosen, and talk about the difficulties that can arise if good reference samples aren't present. So matched reference samples are chosen for each sample of interest. And these controls are chosen based on how well they match the sample of interest. That is, samples with the lowest percent difference are selected to be reference samples for a given sample of interest. We've noticed that performance can be affected pretty significantly if the reference samples selected don't match the coverage profile of the sample of interest. And so if this percent difference on average exceeds a certain amount, you'll start getting pretty low quality CMV calls. And so what we do is we flag samples if the average percent difference is above a certain amount, specifically 20%. So finally, I would like to talk a little bit about the requirements for the algorithm. So first, we do require that you have 100x coverage or more on average to get reliable CMV calls from this algorithm. As far as reference samples go, we recommend that you have at least 30 reference samples. The algorithm does require a minimum of 10, so you can't actually run the algorithm if you have fewer than 10 reference samples. The algorithm also assumes that these samples are going to be from the same platform and be prepared using the same library preparation. And finally, if you're calling CNVs in non-autosomal regions, then you will need to have gender-matched references. And so, for example, if you're trying to call CNVs in the X or the Y chromosome, then it's expected that you would have, at least the minimum of 10 reference samples of the same gender in order for these calls to be performed. So that concludes this brief overview of CNV calling, our motivations behind it, and our approach to the problem. Let's go ahead and jump right into VARSeq and examine how these CNV calls look within VARSeq. So what you're seeing right now is an instance of VARSeq over which we've already called CNVs. So what you'll see down here at the bottom is a table. This is our table of CNV calls. And it includes all of the information about the CNV, the region the CNV is in, the number of targets, how large the region is, and the evidence for that event, as well as the event state. Above this table, we've actually plotted first the actual CNV calls, and so we can see, this het deletion here, and we can see the region that it spans here in Genome Browse. We've also plotted the evidence for these CNVs. So we've plotted the ratio here and the z-score here. And what we're looking at here is a 7-target heterozygous deletion that spans about 19 kilobases. And if we look at this event, we can see that there's pretty strong evidence for a heterozygous deletion at this location. You'll notice that each of these targets have values of about 0.5 for the ratio. And as I mentioned before, this is the exact value you would expect to get for a heterozygous deletion. But beyond that, we see a pretty large drop in terms of the z-score. If you look at the normal regions in this gene here in BRCA1, you see values of the z-score, pretty close to 0. Sometimes we'll get, maybe 1 standard deviation away in either direction. But as soon as we reach this event, we see a significant drop in this z-score, where these particular exons have a z-score that appears to be pretty close to 5 standard deviations away from your reference samples. So we have pretty strong evidence of this heterozygous deletion in this case, and we can see the evidence that was used by the algorithm to call it. So let's look at another sample. So here I'm going to go ahead and look at this sample 12. And you'll notice that our plot updates as we change to this different sample. And you'll notice that our table has changed as well. And so now we have a different heterozygous deletion that we can look at. So let's examine this. So here we have a heterozygous deletion in BRCA2. Again, we have strong evidence for this deletion. We've got a ratio of about 0.5, exactly what we would expect for a heterozygous deletion. And we have a large deviation from our reference samples. Pretty close again to 5 standard deviations away from the normal as defined by our reference samples. But the interesting thing to note about this heterozygous deletion is that this is a deletion of a single exon. This is a heterozygous deletion of exon 7 of BRCA2. And this is a fairly small exon, and so this event only spans 154 base pairs. And it's worth noting that using traditional CNV calling approaches, such as chromosomal microarrays, you wouldn't be able to call this event. A CMA would not be able to pick up a heterozygous deletion of this size, but our algorithm is capable of picking up small events, such as these. Okay, finally, I'd like to look at sample 13, because there's a couple of interesting events here in this particular sample. You'll notice here in our table we've called 2 duplications. So let's look at these. First duplication is in BRCA2, and it spans 4 exons. As with our heterozygous deletion, we see a change in both our ratio and z-score here, indicating a duplication. But in this case, the change is in the opposite direction. So if we look at the z-score, we'll see in the normal regions values very close to 0, and then once we reach the event, we see a large spike in the z-score. In this case, it's around 3 standard deviations away from our reference samples. Likewise, you see a spike in the ratio as well. We've got ratio values that are very close to 1.5, which is the exact ratio we would expect for a copy number 3 duplication. So we've got strong evidence for an event here, and we've called a duplication. Let's look at this other duplication that's in the same sample. This duplication is in PMS2. But there's a few things different about this duplication. Yes, again, we do see a spike in the ratio and a spike in the z-score. But the first thing to notice is that in our table, we've actually flagged this event. We've flagged it as high control variation. This means that if you look at the controls at this particular region, you'll notice that the normalized coverage fluctuates wildly from sample to sample. And that's what's made us flag this event. As a result, you'd expect that these regions would have a pretty high standard deviation. And for those of you that know statistics, a high standard deviation will actually cause our z-score to drop. And that's what we see here. We see a much lower z-score for this duplication event than we've seen in these other events. We see a z-score of about around 1.6, 1.8. And ultimately, given the flag and given the very low z-score that we see here compared to what we would expect an event to have, this is likely to be an unreliable call. And if we had filtered out our flagged variants, this particular variant would have been filtered out and removed. Generally, as a rule of thumb, you would expect values here of at least 3 standard deviations away from the normal. And in fact, if you look at the literature, that's what most algorithms that use simple thresholding approaches do. They'll look and see if the z-score exceeds or falls below 3 standard deviations away from the normal and call their events based on that. And so this value of 1.6 here is definitely a red flag, especially combined with our flags we've thrown here. So that gives you an idea of what these events look like, how you can visualize them in genome browse, and how you can get an idea of the quality of these calls. But there's another feature that we're actively working on right now, and that's the ability to report on CNV events. So if I click the plus button here, I can open up our reporting tab. And in this tab, you can see that we have all of the information related to our report. We can enter in patient information, sample information, all the things you would normally include in a report. We've also included a number of variants here. But you'll notice now there's a location in our report to select a CNV set. And this allows us to include CNVs within our report. Down here in our CNV table, you'll notice we have these red checkboxes. And these checkboxes define a CNV set. And this is just a collection of CNVs that you can create to include them in a report. And to create a CNV set, all we would have to do is click this square button here and click Create CNV Set. So looking at this first duplication, since it was flagged and it has a low average z-score, we probably don't want to include this in our report. But looking at this next duplication, it seems like it has pretty good evidence. It has a very good average z-score. It's not flagged, so we'll go ahead and select it for our CNV set. Once we do that, we can go ahead and select our reported CNVs and include the CNV in the report. And you'll see now we've included this 4-exon duplication in BRCA2. Any of this information can be changed. So for example, maybe we want to say that this is likely pathogenic rather than pathogenic. And once you've entered in your information, you can actually generate your report. So if I click this button, it'll go ahead and generate our report. And you can see the information about the lab, information about the patient, but most importantly, you'll notice that here in our list of affected genes, we've now highlighted BRCA2 to indicate the presence of this duplication that is located in BRCA2. And we've also included the CNV event in our report. You'll see that we've included a CNV event, which is a 4-exon duplication, spanning about 8,000 base pairs, and we've classified it as likely pathogenic. So that gives you an idea of what these CNV events would look like if you include them in the report and how that can be done. The final thing I want to touch on is how you can actually perform CNV calling and go from an empty varseq project to a set of called CNVs. So what you're seeing on my screen now is just an empty varseq project. This is a varseq project with no templates and no variants. So we're going to start out by importing our variants. I'm just going to click Add Files, select our VCF files, and then I'm going to click Next. At this point, we can associate BAM files with our VCFs. And to do that, I'm just going to select Associate BAM File. If the BAM files are in the same folder as your VCFs and the sample names match, it'll automatically associate your BAM files for you. If not, you can click this Browse button and navigate to the folder containing your BAM files. And as I mentioned before, these BAM files will later be used to compute coverage data, and that coverage data will be used to call our CNVs. I'll go ahead and click Finished. And you'll see that our variants have been imported into varseq. Next, I'm going to go ahead and actually compute coverage. To do this, I just click Add, Computed Data, and I'll select the Targeted Region Coverage algorithm. This algorithm does require a bed file that specifies your target regions over which to compute coverage. So I'm going to go ahead and select my bed file. And then I'm going to go ahead and compute coverage. So this algorithm is going to give us a number of useful pieces of information. First, it's going to give us information about the mean depth for all of our coverage regions. It'll also give us information about the minimum depth, the maximum depth, and the percentage covered at each coverage level. It's also worth mentioning this information will also be present at the sample level. And so we can get an idea of the percentage of each sample that's covered at a given coverage threshold. And this can allow us to evaluate sample quality. Now that we've computed coverage, we can go ahead and call our CNVs. To do this, I'm going to again click Add, Computed Data, and I'm going to select our CNV caller algorithm. This is the window that you'll be greeted with when you first select the CNV algorithm. And there's a number of parameters here that can be tuned. The vast majority of these parameters you will never have to change. They've been set to very good default values, but we like to give our users complete control. One of the more useful parameters is this expected CNV rate. If we select an expected CNV rate of not common, the algorithm will call fewer CNV events, and we'll get fewer false positives, but it is possible that this could cause us to miss some true events. Likewise, if we set this value to common, then we will get more CNV calls, but we will likely get more false positives. And ultimately, what this allows you to do is tune between sensitivity and specificity, with the middle value of somewhat common giving a good compromise. You can also specify the maximum number of reference samples used for each of your samples of interest. The recommended value here is 30, but you can go as low as 10. It should be mentioned that it's important that the first time you run this algorithm, you have an appropriate number of reference samples. That is, if, for example, we use the default value of 30 here, then your first project over which you call CNVs should have 31 total samples in the project. That is your sample of interest and the 30 samples to be used as your reference samples. By default, the algorithm will exclude reference samples that have a percent difference greater than a certain threshold. By default, this threshold is 20. So, for example, if we've, say, added 10 reference samples, and then when looking at our 11th reference sample, we see that the percent difference is, say, 25, then we will go ahead and stop adding reference samples and go ahead and perform CNV calling just on those first 10 that we've selected. This option can be disabled by clicking this check mark here. We can also choose whether or not we want to add the samples in the project to the reference set. The first time you run this algorithm, you'll definitely want to add your samples to the reference set. Once we've tuned our parameters, we can click OK, and the CNV calling algorithm will run. The first table you'll see is this new CNV table that's been added. This gives you all the information about your CNVs, information about the region, the number of targets, how large the event is. And you can filter on any of these columns simply by right-clicking the top of the column and selecting Query Column Values. One of the filters I like to apply is a filter to only look at CNVs that are present in the current sample. And so that's what I've done here. I've selected only CNVs for which there's a CNV state of deletion or duplication for the sample of interest. You can also plot any of this information in Genome Browse. So, for example, if I right-click the CNV state column and select Plot for Current Sample, a Genome Browse window will open, and we will see a plot of all of our CNV calls. Likewise, if we go back to our coverage region table, we can plot any of the information in this table as well. And I find that it's very useful to actually look at the evidence for each of our CNV calls. And we can do this by plotting the z-score and plotting our ratio. And so again, I'm just selecting the column with the value I want to plot and selecting plot for current sample. Finally, these plots can be customized pretty easily in Genome Browse. And so what I'm going to do is just add a connector to make these a little easier to view. And at this point, we're ready to look at our CNVs. And so, for example, in our first sample, we can see that we've got a heterozygous deletion call here in APC. And we see a really nice drop in our ratio and a nice drop in our z-score. We are around 8 standard deviations away from our reference samples, and we have a ratio value of about 0.5, which is exactly what we would expect for a heterozygous deletion. And you'll notice that if we click through our samples here, these plots will automatically update to reflect the current sample that you're looking at. And if we go into our table, we can click on any of these values, and we can view the events and analyze them. So, for example, here we can look at this duplication, and we can verify that it's a high-quality duplication. And this one looks like it is, with z-scores indicating that it's between 8 and 11 standard deviations away from the normal, and ratios falling right in that 1.4, 1.5 range. The final thing that I'd like to mention is that we also provide you information about CNVs at the sample level as well. And so if we look at our sample table here, we'll see that we have all the information about our samples, all the coverage information that was computed for our samples. But we also have copy number information associated with the samples. This will include any quality flags warning you if a sample is of low quality for one reason or another. We list the number of events that were called for each sample. We give you information about the spread of the data via the interquartile ranges for the z-score and ratio. But perhaps the most important piece of information here is the percent difference and reference samples columns. The percent difference column tells you the average percent difference for your reference samples when compared to your sample of interest. And so, for example, here, this sample is on average 7% different from the collection of reference samples. And if you start seeing values here get fairly large, approaching 20 or exceeding 20%, then that would set off a red flag. That should indicate that the CNV calls there will likely be of lower quality if you don't have a good match to these reference samples. And you can also look at the individual reference samples that were used to call CNVs for each of these samples. So here on the left, we have the details for our specific sample. And if we scroll down and look at these details, we'll see that we have our list of reference samples here. And we can actually see which specific samples were used as reference samples, along with their percent difference to our sample of interest. So that gives you an overview of the CNV calling algorithm, how it works within VARSeq, and how you can actually go about calling CNVs from your BAM files. If there are any questions, you can feel free to email info@goldenhelix.com, and if you'd like an evaluation of the software, you can do that via www.goldenhelix.com.
Thank you, Nathan. We do have quite a few questions, so we can take just a handful of them here. If you have questions, feel free to enter them in now. If we don't get to you, we will circle back to you after the webcast. I will have the webcast recording and slides up on our website today or tomorrow, and I'll email them out definitely on Friday, so you can keep an eye on your inbox for those. Our next webcast will be on Wednesday, January 11th, and that will discuss imputation. Invitations will go out for that in the next couple of weeks. So let's take some questions. First one, can you tell us about your roadmap for CNV analysis?
Sure, so I can't tell you everything. Obviously, we want to keep a few secrets until we're ready for prime time. However, what I can tell you is that we're currently looking at CMV calling for exome analysis, and we're also exploring runs of homozygosity, among other things.
How is CNV licensed?
So CNV is accessible for VARSeq users. So you're going to need to have a license for VARSeq in order to use it. We then charge an additional license fee for CNV analysis. There are, however, currently running promotions for this, so please reach out to us, talk to one of our area directors, and we'll give you more information about that.
Thank you. How many samples would be required to build a reference set?
So by default, the bare minimum you would probably want is 30. That's what we recommend, is that you have at least 30 reference samples. And if these samples are representative of your sample of interest, you should get quite good CMV calls. The bare minimum that we require to run the algorithm is 10. But again, 30 is definitely the recommended amount.
Is the 100x coverage a requirement or recommendation? Will the algorithm work with 70x coverage, trusting calls at your own risk?
That's exactly what I would say. You can definitely run the algorithm at a lower level of coverage, but the quality of your calls are going to go down as your coverage goes down. And so I would definitely be more wary of CMV calls on a lower coverage panel.
What is the minimum deletion size that can be detected by CNV analysis?
That's an excellent question. Ultimately, the smallest that can be detected is defined by the size of your targets. The smallest event we've personally detected was around the 80 to 90 base pair range, but we can definitely get down to the tens of base pairs at the very smallest.
Do you have performance values for sensitivity and false positive rates at various size thresholds and/or ratio z-score thresholds?
So we do. In fact, our previous webcast would probably be a great resource for finding that information, because we did actually cover that information in our initial CNV webcast. We talk about the precision and the sensitivity that we get on the gene panels that we've run. As a short answer to the question, we've gotten over 90% sensitivity and precision on the gene panels that we've looked at here internally.
Last question here, I think. What do you mean by normalized coverage?
So that's a great question. So what we do to normalize the coverage is actually fairly simple. We take an average of the coverage for your sample of interest across all targets, and then for each target, we compute the normalized coverage by taking the coverage at that target region and dividing it by the average coverage. There's a few other steps that we do to do some data correction, but ultimately that's the process for our normalization.
Thank you very much, Nathan. Nathan did mention further up in the questions that we do have some special pricing on CNV analysis right now. We also have some others as well, but CNV specifically, through December 21st, we are offering our CNV package, which includes 1 seat of VARSeq as well as access to the CNV capabilities for $69.95. If you're interested in exploring VARSeq for your CNV analysis or otherwise, please feel free to reach us. You can email us at info@goldenhelix.com. You can also find more information on our website. And I'm going to wrap up for today. We didn't really scrape the surface on questions, but we will get back to you with answers, so fear not. Until next time, Nathan, thank you for an excellent presentation, and I hope everyone enjoys the rest of their day.